let's talk a bit about kind of empirical measurement. So, today we're going to be measuring people's, how high they can jump. You're going to be measuring people's heights and their calf circumference and all that. And whenever you are measuring things in the real world, you're going to run into some issues. There's randomness in everything. If you're measuring somebody's jump height, every time you do even if you do your really best to do this jump exactly the same way, you're going to get a slightly different jump height, right? So there is this basic randomness that's always going to be there. And when you're measuring things, what you tend to do is you look at lots of different measurements, and then you hope that you're getting somewhere around the real value. So we have what we call like measures of central tendency. I want to know like what is the real height, but every time you do it, it's actually a little different. So I have lots of values that are not the same, but I'm assuming are kind of clustered around kind of the real answer. What is one of the ways we get our measure of central, what's the way we calculate usually? I know somebody's in statistics because they sent me their statistics homework. So what's one of the basic ways to get a measure of central tendency? Like, where is the center of gravity of all of this?
The standard deviation?
So
The standard deviation is going to be related, but something a little different.
Mean or average?
The average. So this is usually we're going to just use the mean average for this. Take a few measurements and average them, and then hope the real answer is kind of that average is, kind of the real answer, even though each measurement was a little different. Right? And there's different, how do you calculate a mean average?
Add and then divide by the number.
Yeah, exactly. You just add all of the samples and then divide by the number of samples. Right? And there's different kinds of averages. There's, there's median, like a median home price in Marin, that's like, where is that middle point where half the numbers are above and half the house prices are below? That's a different kind of average. There's mode, which is the one that shows up the most or something. But mean average is the one that, most people are familiar with. Just what is the value, add up all the values and divide by the number of values, and that gives you the average. And then standard deviation, that is a measure of dispersion. So we should talk about that. Like, how spread out are your samples around that actual average? This could be just the range of the data, so around the mean. If you're in statistics, you typically, calculate variance and standard deviation and things like that. For our class, if you just think about the range, it actually works out okay, fine too. Just so, like, for instance, let's say I have my experiment where I want to see coffee consumption versus test scores. And I could say, these are the coffee drinkers. These are the non-coffee drinkers. And here's the, average test score. Um, maybe that is my bar graph, right? And we didn't talk about this last time, a bar graph. When do you use a bar graph or a bar chart? What's the,
When you have qualitative data?
yeah, so it's like when you're comparing different groups of things. Right? If everything was numbers, you could use a scatter plot. But here we have coffee drinkers versus non-coffee drinkers. So if you're comparing two different categories, you've got to just use one of these bar charts. So here I have my bar chart, and all else being equal, if I just look at this, I could say like, it looks like in general, the coffee drinkers did better on the test than the non-coffee drinkers. But then I want to know, like, do I actually trust that is real, or is it just kind of randomness? So that's where you need to go and think about that measure of dispersion. How scattered around were the actual data points around that average? So if I look at the range, and I found it looked kind of like this, and I realized that among the coffee drinkers and non-coffee drinkers, there were people who failed, there were people who got 100, and it just happens to be that the average of the coffee drinkers ended up being a little higher, the average of the non-coffee drinkers being a bit lower. But I really don't trust that coffee has anything to do with performance on this test, because the difference between these two means compared to how spread out the actual data was that we used to calculate, it's not really meaningful. Does that make sense? Whereas if I had a different situation, these are, the averages are different, and I look at the dispersion around there and it's very tight. If this is like the range of the data, if I have the averages are pretty different and the range of the data is tightly clustered around those averages, this I'm gonna, I kind of trust that there actually is likely something going on here where people who drink coffee seem to be doing better on average than the people who did not drink coffee. And I kind of trust that there's something real. In this case, sure, my averages were a little different between the coffee group and the non-coffee group, but I don't actually think that that's meaningful information given how spread out the actual test scores were in both groups. So does this make sense? So this is part of being kind of a discerning consumer of information. Like I'm not joking when I say people will present data from their own experiments where there's a little change, they'll magnify this. So let's say, if I'll show you, this is something I've seen. You see, we have, okay, this is like 87, this is 92, but it's really spread out. So they'll do a graph like, average test score, 85, 95, coffee, non-coffee. And not give it context. And it's like, wow, it looks like the coffee group did a lot better than the non-coffee group. And then you go into the real data and it's like, yeah, it was this tiny little change, and the actual data is spread way beyond what that actual difference is. So that's why it's really, you can't just compare the averages. You have to think about how spread out is the data around those averages, or else you are going to run into trouble and you might think something is real when in fact it's just random noise. If you're taking statistics, you know, there are lots of things that you can do to try to get an actual calculation of what is the probability that the difference between these two means is meaningful or not meaningful, you know, statistically significant. Like a student's t-test will take in what is the difference between these averages, what is the standard deviation or the spread around those averages, and then we'll tell you how likely is it that these two groups are actually different from each other or just the random variation in a single population. Again, for our class, you don't need to actually calculate the formal statistics and figure out formal significance, but you do need to be able to think about just, does this make sense? Like if you looked at something where the things are spread out, again, a small difference between the means and a large spread in the data, this is making me really not trust there's anything interesting going on. So just kind of putting that out there, because this will come back as we go through the semester, and just realizing that you don't just look at the initial results, you have to really look at analyze the statistical significance. Like, okay, I got this information, but do I actually trust that it's meaningful and not just due to randomness, because the world is really random. Any questions about that? All righty then. Let me pull up. Whoop, here it is. Accuracy versus precision. I'm going to page display. Share screen. So this is just something I got off of the web, but it's actually mathisfun.com. I put this thing up on your Canvas as well. But this idea of accuracy and precision is useful and will play a role in how we do significant figures, which we're going to be having to deal with today already. So basically, you know, accuracy, how close the measured value is to the true value. This is where we're dealing with the averages, right? I'll get back to that. Precision, how close measured values are to each other. So I could have a scale that is kind of broken and it always weighs 10 pounds more than it actually weighs. And every time I step on it and it's 160 pounds 3 times in a row. So I could say there's very high precision. I keep getting the same value when I weigh myself, but it's not very accurate. It's actually 10 pounds off from what I really weigh. Right? So, I mean, and one of the reasons to think about this too is calibrating your instruments is really important. Just because you read a number off of your scale doesn't mean that it's true. You know, when you do real experiments, you tend to double check. Like, let's take a known weight and put it on the scale and make sure it reads what I expect it to. And if it doesn't, I need to recalibrate. So like this thing here, this is kind of a nice little thing showing this here would be accurate. All of these little like darts are clustering, you know, with the central tendency being the bullseye. This is kind of what we're hoping with our measurements. Every time we take a measurement, we're not actually right on the bullseye, but when you average them all, we actually hopefully are getting a number as pretty close to what the actual value is. You know, this case over here where you have all the darts, this is like a really precise instrument that is also really accurate, giving you numbers really close to your target. But it is possible if you have an uncalibrated instrument that you're getting values that seem to be repeatable. You keep getting the same answer, but they're wrong because your scale is off by 10 pounds or something like that. So it's kind of things to think about when you're taking measurements. Coming back to this idea that we're taking measurements with an instrument and we have to average them to get the actual value, that's where we have to think about significant figures. So I know you've seen these in chemistry. We're going to review them briefly because we'll still be using them in our class here. The basic idea is whenever you measure something, there's a limit to the precision of your instrument, right? So if I'm using, in our heights, we're going to be measuring heights today. We're going to use a ruler that has centimeters. And then it's what are these little subunits in between the centimeters here?
Millimeters.
Millimeters.
Right, so given this ruler here, the closest I can get is to the nearest millimeter. So that's the closest I can measure with this. And you do want to use that. When you're taking a measurement, you don't want to throw away, like, you know, if I'm measuring something and it's like, comes out to here, and I say, yeah, it's about 2. You know, you're throwing something away because you can actually say, no, actually it's 1.8. So let's say I measure something and it's 1.8 centimeters. But let's say I do the same measurement again and again. I have 1.8 centimeters. I measure it again, 1.8 centimeters. And then somebody else measures it and they get 1.7 centimeters. And then I want to take the average. You know, I'll just do this because, so 1.7, 1.7, 1, oh, shit, whoops, whoop, shoot, I mean, 1.8, 1.8, 1.8, 1.7, 3 divided by, average is 1.766, you know, and people will write, well, it looks like the length of this thing is 1.767 centimeters. Can you say that?
No.
No, that is not a responsible answer for the average of these real world measurements. Each of these measurements is limited by the precision of our ruler. The ruler has to the nearest millimeter, and basically whenever you have this, you're basically saying this is 1.8 plus or minus, you know, 0.05. So basically it's somewhere between 1.85 and 1.75. It's in between there someplace. That's what that 1.8 really means. So when you get to this, you're only going to be allowed to use the number of significant figures in your least accurate piece of data. So we look over here we only are entitled to 2 significant figures. So we got to round this puppy off here. 1.766, this is going to be, my average is going to be 1.8 centimeters. That is going to be the value I'll use for the length of my doohickey. So in sig figs, you're going to be calculating BMIs using lots of different heights and weights. But you can only use the maximum number of sig figs in the least accurate piece of data you use to calculate that BMI. You can't say, if the height was 167.2 and the weight was, 37.5 kilograms. This is centimeters. You can't say I'm going to make it up. You can't say their BMI is 23.233211. It's got to be rounded off to 3 sig figs because that's your least significant data. So does that all make sense? So keep in mind sig figs as we go through the semester. I should also say always use units. So the units are critical. If you say somebody's height is 167.3, like what? Is it feet? Are they like a giant? Is it millimeters? Are they a leprechaun? You have to say centimeters, for instance. Or somebody weighs 50. Are they like a total scrawny at 50 pounds? Or are they like 100 something if it was kilograms? Or, so you need to be able to always look at the units to know what that number is. There's like this classic example of like this hundreds of $1 million it was a space explorer thing that totally crowds like a satellite. No, it was a space explorer, I think it was a Mars explorer actually, and it totally crashed because it was a collaborative effort between Europeans and Americans, and somebody spazzed and the Americans were using like English units and the Europeans were using metric units, and they didn't realize that they were plugging different numbers into the equations or different units into the equations. So when you actually did all the math, everything was wrong because somebody was saying, oh, this thing weighs 50, and one group thought they meant 50 kilograms and the other group thought they meant 50 pounds, which is, off by a factor of 2. So always use units when you're writing things down, or else you will run into trouble. Speaking of units, this is in your lab notebook. Just review this. If this is not intuitive to you, I know some people know metric units well, particularly if you spent time not in the United States. When I was a little kid, there was all this, I remember in elementary school, we learned this because pretty soon this was, we're all going to SI, but it never happened. And the United States and Britain, to a large part, still uses units. The imperial, in fact, they're even called imperial units. But in science, we always use these. And so it's really critical that you have a good sense of these. So first, the base units, meters for length, grams for weight, liters for volume. And you should have a basic sense of what these are. A meter is about a yard. It's about like, the fish that got away, right? A gram is about a jelly bean. A gram is not very much weight at all. A jelly bean weighs about a gram. A liter is about a quart. About 4 liters is about a gallon. So, if you're not comfortable with, if I'm saying that and you're saying, oh, that's interesting, you should try to make those your own, because you want to have more of an intuitive sense of these units. Because if you don't have an intuitive sense of the units, you can't do that sanity check, right? If you don't actually have an intuition that, oh, a kilogram is about 2.2 pounds, then if I say that this cricket weighs 3 kilograms, you're gonna like, all right, cricket weighs 3 kilograms, rather than like, holy crap, there's no way a cricket weighs 3 kilograms. That'd be like a 7-pound cricket. So you really want to get an intuition if you don't have an intuition. I know in Bio 110, they have a lab where they have you like centimeters about the width of your finger, and decimeters about the width of your hand, and a millimeter is about the width of your fingernail, trying to get you to think of body parts. That's often a good way to think. Like a centimeter is about the width of your finger or so. So, okay, then there are the multipliers. Again, deci, 1/10. Centi, 1/100th. We'll use centimeters a lot. Milli, 1/1000th. We'll talk about millimeters, milliliters, milliosmolar. Whenever you see that word milli, it just means 1/1000th. So if I have 1 milliosmolar, that's the same as 0.001 osmolar. On the other side, things getting bigger, times 1,000 would be kilo. I wrote this little thing at the bottom here. Again, get comfortable with the units and the prefixes and how to interconvert. Interconverting is really easy. It's all multiplying and dividing by 10. You just take the decimal point and you hop it one way or another. Right? And this is another place to do the sanity check. If I have, 100, let's say I have 10 centimeters. And I want to know how many meters is that. So let me change my color here. I can say, okay, if I'm at centimeters, and I want to get up to the base unit, I have to do 2 hops. That's 2 orders of magnitude. So I have to move the decimal point 2 places. And then do I want to move it to the right or to the left? If you don't remember, just do it and see if the answer makes sense. I want to go from centimeters. So 10 centimeters, again, a centimeter is about your finger width. 10 centimeters, about 10 fingers. It's pretty small. I move over, let's move it to the right. I'm going to end up with 1,000, 1,000 meters. Does it make sense that the width of 2 hands is about 1,000 meters, like 2/3 of a mile? No. Okay, that was the wrong direction. Let's move it this way to 0.1 meters. That totally makes sense. A fraction, 1/10 of a meter. So again, you always can use this little sanity check kind of thing. Like, when you convert things and you don't even remember, wait, should I multiply or divide by 100? Just see if your answer makes sense. So just kind of putting that out there. And again, why it's useful. Think about the idea of like a mile. If you're converting between inches and miles, it's ridiculous. It's like 12 inches in a foot, 5,280 feet in a mile. It's just idiotic. Whereas if you're trying to go from centimeters to kilometers, Centimeters is here, a 1, a 2, a 3, a 4, a 5. I take my decimal point and move it by 5 hops, and I'm done. It's so intuitive. So, there's a reason why we use metrics, this system internacional. Let's see. Oh, and I should say, in case people were wondering, what is, it's a joke. So it's a joke. What is 2,000 mockingbirds? So you have to say it out loud.
To Kill a Mockingbird.
Exactly. It's 2 kilo mockingbirds. So that's kind of funny. And so I actually don't have, so here I have nano down to 10 to the minus 9th. I don't know if anybody knows what 10 to the minus 12th is. Does anybody know the prefix for 10 to the minus 12th?
Pico.
Pico. Yeah, so what would 10 of this would be? These would be like peekaboos.
Peekaboos.
But anyway, so those are just jokes though. Those are like, kind of like that atom that loses the electron. There's a lot of kind of nerdy science humor that you can do. So just putting that out there, these units are going to be important. Stop. Share. No, actually, there's one more thing I want to do, actually. I'm going to go back to annotate, clear. So the last, in fact, I am going to stop sharing for a second, and then I will. So over the course of the class, I'm going to be drawing stuff on the board. We're going to be drawing, oh, here's a cell, and here's a channel, and here we have like potassium coming in, and like, there's ATP getting caught to ADP, and there's some sodiums or whatever. I'm going to be drawing things like this on the board. And other times I'm going to be drawing like, here's the heart, and there's the aorta coming out, and there's coming down, we're coming down the legs, and here's somebody's arm or whatever. The scale on the board, they look about the same size, but in reality, the sizes are so radically different. If we look at the size of an atom is somewhere around 10 to the minus 10th meters in diameter, which is also called 1 angstrom. Whereas when we're talking about an entire person, we're talking about, about 1 meter. So this is like 10 orders of magnitude difference here. In fact, 10 factors of 10. So when we think about the different processes in the body, we're going to be thinking about things at the scale of a meter, and also at the very same time thinking about things at the scale of 10 to the minus 10th meters. And just to give you I think an intuition of how radically different it is and what we're actually doing down here. Like in my mind, getting dinky is really not intuitive, but getting bigger is easier for me to visualize. So there's a poem that I think it's a pretty poem, and it talks about orders of magnitude, and I think it's worth kind of going over just because it's giving you a sense of how radically different. So here it's like, Nanao Sakaki. First it sounds kind of like a just a pretty poem, but the more we get into it, you're going to realize it's actually going into orders of magnitude. So within a circle of 1 meter, you sit and pray and sing. All right, that's 1 meter. So instead of getting smaller though, now we're going to get bigger. So now we're going up 1 order of magnitude. You know, now instead of 1, we're at 10. Now you're basically in your room. You know, and you're in a shelter 10 meters large, you sleep well, rain sounds a lullaby. Now we're going up 2 orders of magnitude, 10 to the second, you know, 100. Now you're kind of in a field 100 meters large, you're raising rice and goats. Now we're going to go up 3 orders of magnitude. Within a valley 1,000 meters large, gather firewood, water, wild vegetables, and amanitas. So now we're up at 3 orders of magnitude. Again, we're going to have to get 10 orders of magnitude before we're done here. Within a forest 10 kilometers large, play with raccoons, hawks, poison snakes, and butterflies. So now we are 10 to the 4th. And now we're in kind of a forest even. Again, 10 kilometers, that's a kilometer is about 2/3 of a mile. So this is somewhere around like, you know, 6.5 miles across. Mountainous country, Shinano, 100 kilometers large, where someone lives leisurely, they say. So 100 kilometers, that's 10 to the 5th meters. And now we're in like this whole town, you know, and now within a circle, 10,000 kilometers large, go see the southern coral reef in the summer or the drifting ices in the Sea of Okhotsk. Within a circle 10,000 kilometers large, walking somewhere on the earth. So this is now 10,000 kilometers. So this is 10 to the 7th. Because again, kilometer is order 3, 1,000 is 3, and 10. So now we've got 3 plus 3 plus 1, that's 10 to the 7th. This was the definition originally of a meter. If you take the earth and you go to the equator, from the equator to the pole is 10,000 meters. I mean, 10,000 kilometers, I should say. So 10,000 kilometers is basically the radius of the earth. So at this 10 to the 7th, you are accompanying the entire planet. But we still got 2 more orders of magnitude to go. So within a circle, 100,000 kilometers large, swimming in a sea of shooting stars. So now you're up out of the atmosphere. Within a circle, 1,000,000 kilometers large, upon the spaced out yellowed mustard blossoms, the moon in the east, the sun in the west. We're at 9. Again, million is 6, kilometers is kilos 3. Within a circle 10 billion kilometers large, pop far out of the solar system mandala. That's fine, this is 10, right? A billion is 10 to the 9th plus 10, so that's 10 to the 10th. So to get that same range of size difference, you have to go from you sitting there to popping out of the entire solar system, just to give you a sense of how wide of a range of processes we're looking at. You know, it's kind of amazing that you can actually draw inferences. We are going to be looking at what's happening at the level of individual ions and atoms and see how it's actually having an effect at the level of the entire organism. But just to make you realize, like some of the stuff that we're drawing, I'm going to draw atoms on the board with indiscriminately. Here's an atom moving from here to there. You can't actually see that with your eye. You actually can, you can get information that that's got to be what's happening, but it's beyond what is resolvable with light. So, you know, just because I'm drawing pictures or we'll see simulations of things moving around, that doesn't mean you can actually just get a powerful microscope and see it all. So anyway. So I just want to put that out there so you kind of think, you know, and it obviously gets bigger and bigger. But anyway, so this is also in your, I kind of put in your, where is it? I put it in your one of the modules. I think it's in the module for this lab for today. Okay. Nope. Nope. Do. Right. So if we get to our, oh, did it? Dagnabbit. All right, hold on. I've been trying to get our lab together, and okay, upload, choose file. I've been trying to upload this file, and it's okay. We can do it in a few moments while I'm showing you some stuff. I'm not sure why this is having trouble. All right, so for today's lab, it's going to be kind of similar to what we did last time, which is going to be you watching us do stuff. I was trying to think about whether it made sense for you to try to do the jumps at home, but you know, most ceilings aren't big enough even for, and plus then you'd have to like risk making marks all over your walls. And most of these, you need other people to measure things on you. So I kind of made a judgment call. Like if you want to get your own data and send it in, we can actually add it into the big list, the big data set that we're going to be analyzing next class. But for all else being equal, if you don't do your own measurements, you'll still have everything you need to do the lab. So I'm going to cancel this for now and figure out why it's so. Oh. All right, save and publish. So this should now all be up there. So doing the jumping lab. So I'm gonna play a little video right now. Actually, before we play the video, I want to talk a little bit about the objectives and the hypotheses. In fact, let me stop the sharing one more time. So I have gone through and given everybody feedback. A lot of people are actually looking good at this point. I have my little thing here. I've got, yeah, the majority of the people actually have their objective and their hypotheses looking solid. In general, this is going to be how our labs, these hypothesis-driven labs are going to go. You will have to come up with an objective and a hypothesis, and you'll submit it. And I will give you feedback. And maybe it'll say like, looks great. Probably good chance it won't always be looks great. Sometimes it'll be it's a good start, except there's something you got to fix here. That's normal. I try to remind people that if you were like got everything perfect, you probably shouldn't be in this class. The whole point of being in this class is you are learning skills that you are kind of practicing. And one of those skills is really focusing in how to ask this question in a very explicit way and coming up with a reasonable hypothesis that's clearly stated. So, and one of the things that's sad for me is I have boxes of really cool stamps. Like, and normally when it looks good, I give you a stamp, and it's kind of fun for me and it's fun for everybody. And I was like trying to work, yes, last night I was trying to see if there was some way I'm still gonna try to figure it out, but so far you're just gonna have to be satisfied with me saying looks good or something to know that it's approved. But when we do the, what's the next lab? We do the next labs that are also going to be like this. You're going to be, what is the Osmometer Lab? The Osmometer Lab is going to have a hypothesis and objective, and I'll have you submit it. again, for 0 points, and I'll give you feedback, and there'll be an iterative process. And the point there is just before we actually do the lab, I feel comfortable that everybody knows what is the question we're asking and that you've thought it through enough to come up with some idea of what you think you might find. So that's that. And most people got the idea of the objective. There's one person that still didn't kind of get the idea that it is the independent and dependent variable. Most people did get that. It has to be all the independent and dependent variables. So like what would be a good statement of the objective for today's lab?
Determine if a person's height, weight, BMI, and calf size affect the height of their jump.
Yeah, exactly. Something to that effect. The main variables we're measuring, their height, their calf circumference, and their weight. And is that correlated with their ability, their vertical jump? You can throw in BMI or not for the in the objective. It doesn't matter as much in the objective here because it falls out of the other variables that we are looking at. So that might come up in your hypothesis as a combination of the independent variables or in your analysis later. And what about hypotheses? And again, there's no correct hypothesis. There's lots of possible hypotheses. Does anyone want to share a hypothesis?
I could.
Okay.
I could. Okay. If height is directly proportional with an individual's ability to vertically jump, then the taller the individual, the larger the ability to vertically jump, because the longer the muscle, the larger the potential energy that can be used when jumping.
All right, so she's saying like taller people will be able to jump higher, and then she has a good reason. Like people who are taller are going to have physically larger muscles, which you expect going to be able to generate more force to be able to jump higher. So that's the kind of thing you want. You want what you expect, and then a reasonable explanation of why that makes sense. Some people made a bit, I think because maybe they didn't understand the procedure as well, said taller people will jump higher because they're already reaching up higher to start with. And that would not be a good hypothesis, because in our methodology, you're going to have the subject mark the wall at whatever height they're at. And then you see how high can they jump above that. So a short person or a tall person could still have a 2-foot jump. It doesn't matter how high absolutely off the ground they are. What matters is how high can they jump above their kind of baseline height. So if you had that in your hypothesis, I kind of commented it in the comments. If you haven't fixed that yet, make sure you do fix that. And how about somebody else has a different hypothesis?
Could it be like shorter people jump higher because they have lower body weight?
If it's about lower body weight, then why don't you just use weight versus jump? You could say lighter people jump higher because they have less weight to lift off the ground, maybe. Right, a shorter person doesn't necessarily have lower body weight. A shorter person could be, short person could be a string bean or could be a fire plug, right? You don't know about weight. You can have, in fact, I can say, yeah, I can say I know people with very different heights that have the exact same weight. You know, in fact, that's where you kind of go more with BMI. BMI is the thing that says for the same height, you can have people that weigh quite a bit more, quite a bit less. So,
Hello?
yeah. Well, now I hear.
I put my hypothesis about the weights because the people have low weights, they have more they're less obese and then have a probability to jump higher than the other ones.
Right. So that would be another possible answers or possible hypothesis. So people who there's going to be a inverse correlation between weight and jump height because less weight means, you know, less fighting gravity and easier to get off the ground. So that would be another possible hypothesis. What about another one?
Or the people that have been like doing like run, don't have more training, doing more exercise.
Right. So that could be an interesting question, but that is not something we're actually measuring. So in your hypothesis, you want to make sure that you are limiting yourself to the actual variables that we are looking at. So your hypothesis has to be relating directly to the independent and dependent variables from the objective. Okay. So, you know, those could be interesting. Those are probably sources of variability for our experiment, but they wouldn't be appropriate in your hypothesis.
Okay, thank you.
I know there's other ones because I read them. They need people to just.
I said for that those with greater calf circumference might jump higher because they would have more muscles in their calf to generate a greater force and be able to jump higher.
So there's a third possibility that is also completely reasonable. Larger calf circumference, you know, likely due to larger calf muscle to generate force, you know, with that plantar flexion with your gastrocnemius or whatever. So those are, you know, just part of the reason I'm kind of going through this is just to make you realize there's no right answer. There's lots of possible things, and there could be more than one right answer, or could be none of them have any correlation. We're not going to know until we look. And so for our first analysis, we're going to go through the actual kind of experiment, and then I'm going to talk about initial analysis. For your initial analysis, I just want people to stay focused on your own hypotheses. You know, that's usually the first part of any discussion is, you know, does the data support my hypothesis or not? You know, next week, we are going to go deeper into the analysis. We're going to get larger data sets across many different labs. We're going to look at all, not just look at your hypothesis, but, you know, we got the data. Let's check out height and weight and calf circumference and sex and whatever else to see if, because we're also recording whether it's a male or female jumping. And we can then see if there's something interesting in there that maybe we didn't expect. The reality is, even though, you know, humans, we're clever monkeys, we can figure things out and try to figure what are we going to find. Often the most interesting things are something you never figured to find in the first place. That happens a lot. You get your data and you start you definitely see as your hypothesis supported, but then you double check, well, maybe my hypothesis isn't supported, but there is something else interesting going on that's worth paying attention to. That is a very common thing in science, is you notice something that you weren't expecting, and then you use that as your new hypothesis to explore. So I don't want you to think that the whole process is so rigidly laid out. You know, parts of it are, but a lot of it happens to be like you noticed something interesting in the world. You know, serendipity, the whole classic will talk about how, the way people discovered penicillin or lysozyme, the antimicrobial stuff in your snot, happened just because people were being observant and noticed something they weren't expecting. So, all righty. So I am going to go and start this little video of doing the jumping lab, and I'll break in as appropriate. Introduction and body measurements. Yeah, this is online too. This is in the module. This is the jumping lab, and in this lab we're taking a variety of measurements off of our subjects, and then looking for correlations with their jumping height. So one of the measurements we're going to be taking is their height. Again, just talking about sources of variability, your height is going to be really different depending if you're wearing platform shoes or you're barefoot. So before you take anybody's height, you got to make sure they don't got shoes on. So no shoes. Another thing, some people, I don't have poofy hair, obviously. Some people have poofier hair. You want to make sure as you're measuring height that you compress down and you're actually getting their actual height and not the height of their. So here we have meter sticks taped on the wall. I'm going to stand here straight and let's see.
Okay.
I'm going to show parallax.
What was that?
Show parallax. Like, you have to make sure it's parallel if you can over or underestimate that thing.
Oh, yeah. So it's important that it's parallel or perpendicular to the ruler in the wall, because you could measure too high by doing this or too low by doing this. So you want to have it completely perpendicular, and these meter sticks are marked.
So so that is a big issue. Like for right now, Tina is tall enough that she can actually measure my height accurately and notice whether the ruler is parallel. But sometimes you end up having kind of a shorter person trying to measure, and they don't even know that the ruler is up at an angle and is actually reading a centimeter higher than it should. So if you're measuring somebody's height and you don't have a good view of the top of the ruler, it's good to step on a little bench or step on a little stool to make sure that that angle is just right. Because it's called parallax error, when you're looking at it from a non-parallel angle and you get a bad reading. So that's something to always pay attention to. It's also important. I'm just going to say, we have these 2 meter sticks taped on top of each other. This is where, again, that sanity check. Again, the first one is 1 meter. But then people will read like, oh, they'll just read 67 off of this. And it should be, well, there's 100 centimeters down up to the first meter stick. So now we're at 167. But there'll be 4 people in the group and they'll all just agree, oh, this person's height is 67 centimeters, right? And again, that's where if you know, like, 67 centimeters, that's less than a meter. 100 centimeters is a meter. So that's like 2 feet. But I'll have like entire groups of people in this lab that are just all like, okay, let's write down, if they were writing down that their subject is 2 feet tall, everybody would say, that's ridiculous, something's wrong. But when we're doing it in centimeters, people aren't used to, they don't have an intuition. So, and that's why I'm saying, please spend time. If you're not comfortable with the metric units, get comfortable. Know that 60 centimeters would never be a good height for a human being, or an adult human being, I should say. It could be okay for a baby or something. All right.
Too low by doing this. So you want to have it completely perpendicular. And these meter sticks are marked with millimeter tick marks. So in this case, we want to measure to the nearest millimeter. And then you want to record that in your lab notebook. And also don't forget that there are 2 meter sticks taped here. So any person in this lab is definitely going to be over 1 meter tall. And then we reach to the nearest millimeter here. So in this case, it could be 1.6658. And then you can convert that over to meters.
And in the actual lab, you would have more than one person take the same measurements, because even though it seems like a straightforward thing, there are so many slight variations, the exact angle of the meter stick, or just somebody's approximation of whether it's up or down 1 millimeter, that can actually lead to a different value. So to try to get at the true value, we take multiple measurements and we can then average them and then assume that that average of those different measurements is approaching our true height. All right, so this next part is measuring the cap circumference. And again, here's another example where we need to bring in this idea of thinking about sources of variability before you actually do the experiment. Like, just like you have right-handed and left-handed people, there are people who are right-legged and left-legged, have a leg that they prefer. And they're going to probably have more muscles on their leg that they prefer. So the calves are probably not exactly the same. So it actually is going to be important to acknowledge that before we start, and then come up with some way to deal with that. And the way we'll deal with it today is we'll measure the calf circumference of their dominant leg. That would be the leg, like if you were told, go hop on 1 foot from this side of the room to the other side of the room, which leg do you hop on? That is your dominant leg. So when your subject is getting ready for this, have them hop and see which leg they prefer, and then you want to measure that one. Also, I highly recommend, if you know you're taking your shoes off for this lab, wear fun socks.
All right, 37. And we're going to measure it 3 times with 3 different people measuring the subject's cap circumference, and then you're going to average that.
Right. So, and notice just being slightly up or down is going to change the actual calf circumference, right? So this is, or if you're pulling a little tighter or a little less tight and compressing the flesh a little more or less, that's going to change the actual millimeters of the calf. So that's why we have like 3 different people do the exact same measurement, and probably they're all going to make slightly different mistakes or slightly different, you know, up or down or tight or loose. And we're hoping that the average of all 3 measurements ends up becoming the, you know, close to the true measurement. And for the weight, we just have people step on a scale. That one's easy. So you don't have to, we didn't actually make a video of me stepping on a scale. So before we continue with the actual jump, there it is. One thing I also want to address from your pre-labs are the sources of variability. What were sources of variability that people came up with?
Genetics.
So genetics can definitely make a difference. And again, that's something we can't control.
Injuries.
Yeah. Level of energy, if the person is tired or not.
The level of
training.
Level of training, you know, and again, things like level of energy we can't help. What we can help is kind of attitude. In fact, in sports psychology, people talk about visualization and really trying to get your head behind what you're doing. It does make a difference if someone's like, oh, this is kind of embarrassing and I can't jump. Okay, let's do this. Versus, all right, let's do it. Jump. You know, then it makes, in fact, we actually, in this semester, we're not going to do the independent research projects because it just doesn't make sense. We're doing instead we're doing a different research project. But we've had students in the past do experiments where they take the jumping lab and they have people do these little affirmations and say, you know, like, I feel like lead, I can't jump, this is stupid, that's like blah, blah, blah. And then they'll try to do their jump and they'll have other people like, you know, I'm a great jumper and I'm full of energy and I'm gonna reach for the sky or whatever. And, you know, it really seems to correlate with jump height. So having a good attitude makes a difference. And again, if we were doing this in real life, I would be encouraging everybody to kind of don't feel embarrassed about jumping because, you know, everybody's jumping. But just kind of putting that out there. Source of variability is even your attitude. And that one we can control for. You know, and other things we can control for. Someone had said training. You know, we can control for at least a modicum of that by making sure everybody is trained to do the same jump and then doing the jump in the same way. We can control for, you know, height and calf due to shoes. Like if somebody's wearing high heels and you measure their calf, high heels makes your calf muscle tense up, which is going to change your calf circumference. So we make sure everybody is getting their calf measured, standing flat-footed on the ground. And we're making sure that you're measuring your height without wearing any shoes and doing the jump without any like springy like basketball shoes on. So, you know, in general, it's really important to think about these sources of variability so you can figure out how to avoid problems with them as you're doing the experiment if possible. Any other sources of variability people came up with?
The level, okay, I'm sorry. The level of energy.
And so yeah, again, level of energy, or even what, you know, again, if they drank Red Bull before class, or whether they took some Valium before they came to class, or had a beer or something.
Body proportion.
So, I mean, it could be body proportion. We're talking about just their height. I mean, proportion, in some degree, that's going to come out in like BMI to some degree. But then there's other things about the person with the same height could have really long legs or shorter legs. Like I have one of my colleagues who's quite a bit taller than me is got this from his feet to his waist is exactly the same height as me. It's bizarre. But then from his waist to his head, he's like a foot taller. So different people are stretched out differently. And that, I'm sure that makes a big difference with the lever arms and stuff as you're generating force. So what we're gonna do now is actually show you what the jump looks like. And I'm gonna go back into sharing. All right, so measure my height, my calf circumference multiple times so we get hopefully as true a value as possible. We've also measured my weight standing on a scale. You all know how to do that. And now we're going to talk about our dependent variable in this whole experiment, the actual vertical jump height that I'm going to be able to do. A few things before we start that. Another source of variability is the actual mechanics of the jump itself. If you just kind of like that or whatever, it's going to make a big difference on how high you jump. And because we want to look across many subjects, we want to have a standard jump where everybody's doing the same mechanics, which I'll briefly show you. It's basically as described in your lab manual. You standing vertically, your hands by your side, you bend your knees, and then you're going to extend your knees as you swing your arms upward. And you want to have your legs and your arms extending simultaneously so they both are at full extension at the same time. So there's this really nice kind of synergy that happens. And you also want to make sure you're using both arms. A lot of people have this tendency to like jump like that and just throw up one arm and they have their half of their body is just kind of paralyzed. So you want to have your body straight, bending at your knees, and then in one smooth motion, explode up with your arms going up, legs straightening, and actually show how it's done. I'm going to have my pencil here where I'm actually going to be able to mark. You know, obviously, we want to see how high can I jump from my normal height. I don't care if a person is taller or shorter. You know, they're going to be able to mark the paper higher or lower depending on their height. That has nothing to do with their jump. So what I care about is where do they mark it if they're just standing on the ground? And then I'm going to be able to jump and mark it at the peak of my jump. And then I'll see what's that relative distance. And so I'll do that. This is my baseline. I'm flat-footed. Now I'm going to go and do this jump. Bend my legs. Ready? And I want to, again, every time you jump, you're obviously not going to jump exactly the same height. So another thing we're going to do, just like we do multiple measurements for the height and all that, we do multiple measurements for the jump. All about that. And holding the pencil at the exact same angle that I'm going to mark at the top of my jump. I'm going to get ready. Straight body, bent legs. Go up. There's the second jump. And I'll get ready. And one last time. And my lab partner, if I go in like this, my lab partner's got to say like, whoa, that wasn't it's nice to watch you jump, but that wasn't a jump we can count because you didn't have the proper form. So as a person, you know, making sure the experiment is done correctly, you need that, you don't want to throw away data just because it isn't what you thought it should be, but you don't want to take data if they actually didn't do the procedure properly. That's okay, my third jump, bend my knees, stand up. And now I have 3 little marks up there, and now it's time to measure how high my vertical jump is so we can see how it correlates with my height, my weight, my cap circumference, and some other things that will.
All right, so next it's time to measure David's jumps here. So carefully, without falling off, you want to get up here. You use the metric system of any ruler that you have. And then, of course, it's not very handy for me to record this. I'm going to have to yell out the numbers for my lab partner to write down in the lab notebook. So I'm going to go from the baseline to the first mark here at the highest point. And it's going to be 47 centimeters and a half a centimeter. So 47.5 centimeters. 47.5. All right, second jump. It's going to be 46.2 centimeters. 46.2. And the third is 47.0 centimeters. So those are the 3 jumps that they did. All right.
I look silly there. All right. So that is what we would have done. And again, I mean, it's not my fault, but I still kind of feel like apologize that you actually aren't going to all be. It's kind of fun to just jump and do stuff. But instead, what we're going to do is just give you data for this. For some of the other stuff, there are things you can do at home. This one, I think, makes sense just to get the data. And what time is it? It's what I want people to do is do some of the initial analysis to look at what your hypothesis looks like in light of actual data. And so what I need to do is get this file uploaded. For some reason, it was not uploading. So if you could please bear with me. Talk amongst yourselves. This will probably take me a couple of moments to just get this thing up, figure out why it's not uploading properly.
Professor, I didn't get a calf circumference. Did they mention it?
No, it's okay. We're gonna, yeah, you don't need to know that. Yeah. Do, 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 do. Ha. All right. All right, so we have, yeah, I have no idea why it was not uploading, but I managed to kind of go through the back door of the stupid thing and get the file in there. So what I just put into the unit is data from, actually from like 2012. It's just from a class kind of like you were. And it's got the people's height, weight, calf circumference, BMI, jump. And what I want to talk about now is some of the ways that you are going to go about analyzing the data. We've got like a half hour here, so I just want to spend a little bit of time talking about the realities of what your discussion is going to look like as we start this lab. Again, next week, we're going to go much more into depth. On Tuesday, we're going to continue looking at the jumping lab. We're going to have a much larger data set across many classes. We're going to use more advanced graphing capabilities of Excel and all that. But the first step here is going to be the initial analysis. And your discussion, again, is always going to be, you know, here we had a question. Here was our hypothesis. So now that we've got our data, what does our data tell us about our hypothesis? Does it support our hypothesis? Does it not support our hypothesis? And how you are going to answer that depends on your hypothesis. You know, what you want to actually analyze in the data is going to depend on the particular hypothesis you have. So if I go and like share screen here. So this is what the Excel spreadsheet looks like. And typically when you have a big list of things like this, you can just put the units at the top of the column. You don't put the units on every number. You just, the assumption is, you know, everything here is kilograms. Everything here is centimeters. Everything here is centimeters. Everything here is centimeters. So this is my data. So we've talked about like 3 different kinds of hypotheses now. We had one hypothesis that said that somebody's calf circumference was going to be directly correlated with their jump height. So what kind of graph would I use to figure out whether or not their calf circumference is actually correlated with their jump height?
A bar graph to compare?
So the bar graph is when you have different categories of things.
Ah, okay. Okay.
So if a bar graph, if I wanted to tell, like, you know, men versus women, that would be a good reason to use a bar graph. You know, for that, I could just pick all of the men, you know, so I could say, okay, here, let's make them blue. But that's not what I meant to do. Undo. I'm going to say, okay, there. In reality, I'm going to show you much more efficient ways to do this in Excel next week. But for right now, since we're doing this manually, I'm showing you this kind of awkward way to do this here. If I was going to look at men versus women, I could, let's get, the woman will be pink. You know, here I'm just doing this to highlight to make it easier. And I'd get out my calculator here and I could say, okay, for men, we have, you know, 61.2 plus 54.6 plus 40.4 plus 41.8 plus 45.3 plus 43 plus 47, and then divided by 1, 2, 3, 4, 5, 6, 7. 7 divided by. So the average jump for the guy was 47 point. Again, I can, if I go back over to here. What? Hold on, what's going on? So I did my calculation. I got 47.867 centimeters. Can I actually use that all?
No,
No.
What would I actually use for my jump value?
47.9.
47 point.
9.
8.9, because this averages up. So this would be for male. And then for the female, you know, the reality is I think I'm gonna cheat here and use I'm gonna use the averaging function here because I just don't want to bore you all here. So for women, it's 28.4. So this is average jump for men and for women. And if I wanted to show this in a graph, I would just make a bar graph. This would be a perfect reason to make a bar graph. You know, average jump in centimeters. 25, 30, 35, 40, 45, 50. And I could have male or men and women. And I could say men goes up to 47 about, women comes up to 20 about there. So this would be a good reason to use a bar graph where there's two categories, men versus women. If I wanted to be a little more interesting, I could put ranges. If I look at my range here, the men's jump, the smallest jump was 40, the highest jump was 61. So for the men's jump, it goes like from here up to here. For women, my smallest jump is 14 and my tallest jump is 40. You know, so when I look, if this is kind of the range of my data, you know, if I'm looking at this, this is telling me, yeah, on average, men seem to jump higher than women. And just my gut from looking, you know, I could do the more formal statistical analysis, but just from looking at these difference in the averages and the clustering of the data around the averages, I think there's probably something real going on that in general, at least with the samples that we're looking at, men on average jump higher than women. You know, that doesn't mean that any particular man is going to jump higher than any particular woman. There are women that jumped higher than, you know, the lowest guys and guys that, you know, so, but on average, men jumped higher than women. So this would be a good example of a bar chart. Another reason, what would be another thing you could examine using the bar chart that would be relevant for our data? Think about BMI. If you people who were talking about healthy versus unhealthy BMI, I could just take everybody who had a BMI in a healthy range and average their jump, and take all the people with the unhealthy BMI and average their jump, and I could get a comparison of healthy to unhealthy BMI and see if that makes a difference. Right? So whenever you're comparing different groups, you're going to want to use kind of this bar graph, bar chart. What about if you're comparing correlations between quantitative things, like somebody's calf circumference compared to how high they can jump?
Scatter plot.
That's going to be a scatter plot. You usually have the dependent variable on the y-axis, the independent variable on the x-axis. And then before you actually do this, you have to kind of look at the range of the data. So if I go back over here, let me undo my sorting here. If I'm going to do calf versus jump, let me get rid of these colors just to make it. I look at my calves here, and I look at the range. Like there's no reason to start calf at 0 on my graph because nobody has a calf of 0. I want to spread the data out so I can analyze it well. So I can say, all right, calf, the smallest calf I have is like 35. The widest calf I have is, looks like 44. So I'm going to probably have my calves go from 35 to 45 and spread that out along the whole x-axis. And then for the jump, my jumps go everywhere from 14 up to 61. So I'll probably have my jumps going from like, 10 to 60 or so. So that's going to be, before I actually do my graph, I first have to look at the range of the data so I can spread things out. So my calf, I'm writing it down here because it's going to go from about, actually calf, well, there's a calf that's 30.
You have a data point at 31.5 for the calf.
Right, so I'm going to go, so I'll go from 30 to 45 for the calf. And then for the jump, I'll go from, from 10 to 65. All right, so then for a lot of graphs, we will be using Excel. Excel excels at making nice graphs. But I also want you to believe in yourself that you can graph things, because sometimes there's things that are just simple graphs and people get, oh, I got to type it into Excel and graph it. And it's like, just put some dots on a paper and it's easy. As long as you kind of trust you can do it. A lot of people have like math anxiety. So part of what I want you to kind of get past is math anxiety, if that's something that affects you. That's part of the reason why your your lab notebook has graph paper in it. It's to make nice tables and also so you can just do a quick, easy graph if you need to. So I'm actually going to use a ruler, which I'm going to use to actually draw on my iPad because you still want this to be pretty if it's going to be something you get data from. It's going to be easier without the keypad. I'm going to put this flat. Okay, I'm going line this way, line this way. And now I know that I want my calf to go from 30 to 45. I'm actually taking my ruler here Um, half, so let's, so how many, that's 20, that's 25. So if I do, let's let this be 30, 30, 30, let's do 2 each. 2, 2, 4, 6, 8, 10. This is 40. Oh no, what am I saying? No, this could be 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. This is actually 35, 36, 37, 38, 39, 40, 41, 42, 43, 44. Oh, totally spazzing. Sorry, I so apologize. All right. This is my dependent variable here. This has to be the jump height. So I just spazzed on you. Jump height in centimeters. This is going to be the calf in centimeters. So this is going to go from 30 to 45. So this is going to be, let's be 30. 1, 2, 3, 4, 5, 32, 33, 34, 35, 36, 37, 38, 39, 40, 41, 42, 43, 44, 45. So there's my calf circumference on the x-axis. It's important that you don't spaz on the numbering because you're actually going to be using this in an analytical way. So it has to be exact. If this distance from here to here is 1, it has to be 1 consistently everywhere on that same axis. People get all kind of sloppy when they are not used to making these graphs. Jump height has to go from 10 to 65. So let's do that. 10, 15, 20, 25, 30, 35, 40, 45, 50, 55, 60, 65. Notice these have to be also a consistent thing, but it can be a different amount of centimeters per division. It's important though that, again, that is consistent. And now I'm ready to graph. And notice I made my data kind of spread out. So my range of data, which was about 30 to 45 for the calf, and the range of the jump heights, which is about 10 to 65 for the jump heights, it gives me a nice little, like the aspect ratio about this, looks like a TV screen. You don't want to come up with this thing so it's all scrunched and all the data is all in a, you want the data to be spread out so as you put your dots in, it lays out nicely like this. And now, let me just to make my life easier here. And so what do I do next? How do I actually make my scatter plot?
you plot the calf circumference with the height of the jump.
And for what?
And put a dot.
We're gonna put a dot. Every subject is gonna be a dot, as opposed to that scatter plot where the bar is actually the average of a whole bunch of different people. Each dot is gonna be a person. So I'll just start. First person is 35 with a jump of 61. So 35 and the jump of 61. This is the first person is right there. The next one, calf is 44, jump is 27. So calf is 44, jump is 27. That's like right around here. The next one, 37, 40. That's about right here. The next one is 41, 54. Right about here. The next one is 41, 33. Right about here. The next one is 42, 33. The next one is 36, 40. The next one is 39, 20. The next one is 35 and a half, 29. $32 and a half, 37.
Those last two subjects in the right place, they were like 30 something. So wouldn't they be?
I mean, if I messed it up, it's okay for right now.
Okay.
35.6, 21, 35.6, 21, look around there. 34, 32. So you kind of get it. It's just each person ends up being a dot here. 33, 22, I just did that one. 35, 27. 41, 42. 40, 45. 35, 32. 43, 31. 34, 31. 35, 43. I'm almost done. 35, 43. Be up here. I'll skip to the end. 40, 24. So that you get a sense. This is what a scatter plot is. Each character ends up, and then you have to do your best fit line. Try to say, is there a trend? Do these things, obviously it's messy, but does there seem to be a trend? And if I look at this, I would say, I don't really know if I see a trend. The rule of thumb, people are pretty good at eyeballing it. You basically want like the line that's closest enough to all the dots. And in fact, the way that's calculated, the least squares, you basically do a calculation of what is the distance between the dot and the line and calculate which line minimizes the distance from all the different dots. You can also say like, what's the line where half the dots are above it and half the dots are below it? That's also a good rule of thumb. When we get into Excel will just calculate it for you like that. Does there seem to be much of a relationship here between calf circumference and jump height?
No.
No. So if this was your hypothesis, we would say our data doesn't really seem to support the hypothesis, at least with this initial analysis. So for Tuesday, all I want you to do is some initial analysis. I want you to have a graph. I want you to have an initial conclusion, like does the data, this first small data set, seem to support your hypothesis? We are going to do much more detailed analysis on Tuesday, but don't worry about that yet. I just want you to look at your individual hypothesis. If there's one person that had multiple if you have multiple hypotheses, just pick one of them. You don't have to do it for everyone. But you should analyze the data and see, does it support your hypothesis or not? If I was going to say the Cap circumference, but only for the people with healthy BMI, how would I try to figure that out?
Only do a scatter plot of the people with healthy BMIs.
Exactly. So I would take, okay, let's pick the people, BMIs, 31, that's a health, 18 is healthy, 33 is not. Let's just pick the people with healthy BMIs. No, 22, 29 is not, 26 is not. This is a good one again, 23. So just pick the BMIs that are healthy, exclude the subjects with unhealthy BMIs, and just graph the calf versus jump for those people. That would be a way that you could calculate or check calf versus jump, but just for healthy BMI. Just pick the subjects that have a healthy BMI, and then graph the calf for them.
What was the range for healthy BMI, Dr. E?
It's like, I think 18 to 24.9.
Thank you.
So for Tuesday, do an initial analysis of just your hypothesis, just using this limited data set. I am going to put another little upload link for you where you can upload this sometime before Tuesday as a PDF. If you're having trouble figuring out how should you analyze this or how can you graph it, let me know and I'll try to help you with that.
Does this go in our lab notebook?
This does go in your lab notebook. This is going to be like your initial analysis of the jumping lab data. And on Tuesday, we are going to be diving much deeper into having a much bigger data set and how can we start looking at, first we're going to be able to compare. Does your initial results from looking at this limited data set of just this one class, do they still stand up when we look at data across many different sections when you have much larger numbers of subjects, which is an interesting thing to think about of something that looks like it's supported or not supported. Do we still see that same result when we look at more and more subjects, which should be giving us a more realistic result because the sources of variability tend to cancel each other out and real effects tend to float up above the surface. Are there questions about what the expectations are? Maria?
Yes, Professor. When doing the analysis, the initial analysis, do we have to say the why? We didn't have the results that we expected in our hypothesis or not? Just that if the data supported our hypothesis or not?
Right. So you'll never know exactly why or what's going on, but you'll be able to tell whether or not data, your prediction seems to be supported or not.
Okay, thank you.
So that's, so this first part is just, does the data seem to support your hypothesis, yes or no? And then we're gonna continue looking at this jumping lab in much more detail next week. But for right now, I just want you to have this sense of, okay, here's some, I had a hypothesis, here's some real data, and now let me see whether or not the real data seems to support my hypothesis or not. And again, we're going to look at it in more detail later, but this is kind of the initial analyses.
And this is online, the data set?
Yeah, the data set should be available now in that doing the jumping lab page in the module. And I would request, even though it's in Excel, I would request that you start by just doing a hand-drawn graph in your lab notebook, just because I do want you to feel comfortable making a hand-drawn graph. And there's not that many data points for this one, so it's not going to take you that long. It's going to be, it's not going to take you that long. Later on, again, next week, when we're working with hundreds of subjects, then you're going to want to use Excel graphing tools, but I want you to just have a sense, believe in yourself, because I definitely know, again, from teaching this class for many years, that people come in feeling like that graphs are scary, and I want you to kind of feel like, oh, no, it's not. All right, any comments, questions, or anything before we go into office hours?
I had one question. The chem labs due Friday, right?
Yes, the chem lab is due Friday. And what I'm going to do is I'll give people feedback, and for that one as well, give you a chance to revise things. So it's like, turn it in, and then I'll give you feedback by the end of the week. And if it looks like things need to be fixed, just like we were doing with this pre-lab to the jumping lab, especially in the beginning, there's probably going to be a lot of this back and forth of like, that part looks good, but this part actually needs to be developed more completely. So you'll turn it in, but I will, if it looks good, I'll just give you a grade for it. But if it looks like it needs help, I will send it back to you for revision before I actually give it a final grade. So any other comments or questions about that stuff?
Did you want us to have the graph that you just did in our notebook, or was that just an example of how we should do ours?
If your hypothesis has to do with cap circumference, you're going to want to have that graph. If your hypothesis had to do with something else, it'll probably be a different graph.
Okay, thank you.
Yeah, so the graph that you make is going to depend on your specific hypothesis that you're trying to find support for. Yeah, so some of you will have that graph and other ones will not. Other comments, questions? All right, so I guess it's Thursday. So have an awesome weekend. Actually, Labor Day. So yeah, you don't actually get a holiday for this class, but maybe you get holidays from other things in your life. And I will see you all on Tuesday at 8 a.m.
Thank you, Dr. E.
Cool. And I will just stay online here. And if people have questions, I'm happy to talk with you.